Hello and welcome to my presentation, When Synapses Become Sour, the Influence of the Intracellular pH and CO2 on Neurotransmission. This is a project I did as an elective in my faculty, and I'm a veterinary student at the University of Leipzig right now, and I'm in the 8th semester. I participated in a program that is called Rise Weltweit, that is sponsored by the DAD, which is the German Academic Exchange Service, and this is a scholarship that promotes research internships worldwide up to 12 weeks, and if you're in veterinary school, you have to be done with a physicum, which is the preclinical part of the studies, and they have a database basically of projects worldwide where researchers are open to take up an exchange student or an intern from Germany. And I was able to go to the University of Kentucky to the Cooper Lab in the Department of Biology. And momentarily, or in the near future, Felicitas Koch, also a student from the veterinary faculty of Leipzig, is going to go there and continue the research. Furthermore, there is a alumni meeting in Münster every year in November where all the scholars attend, all the students, and it's really interesting to see all the different students, and they also give presentations on their topics. And it's really interesting because it's not only medicine that is being presented, but also mathematics, geography, and such. So it is a lot of fun. And if you have any further questions, please contact me. So first I would like to introduce you to my outline. The first thing I will do is explain the model organisms I used, and then explain some of the physiological background. Then I will continue with the methods, what we did, and then come to the results. First on crayfish, and then also on the fly. And in the very end, I will sum everything up. So the model organisms I used was the crayfish and the Drosophila, and those are invertebrates that are used quite often in research, especially because there have been concerns about using vertebrates, but also because they have been used for a long, long time. And there are a lot of similarities concerning those crayfish and Drosophila and humans, and they've been used since the 1800s. Especially a fly is used in genetics, and its genome is sequenced quite precisely. So you have a lot of different models that match diseases in humans. For example, Alzheimer, Parkinson, and Huntington. So a lot of the genes they have, we do have also, and we can match the neurological diseases with them. So what we did on the crayfish as this preparation, we used the leg and we prepared it to look at the opener muscle because we wanted to look at the potentials in there. So what you can see here is, in the second picture, is the nerve that we took to stimulate it, and in the third picture you can see the preparation of the opener muscle, which is a V-shaped muscle down there. And in the last picture, we stained it with methylene blue, and what you can see is the two branches of the nerve going into the opener muscle. And you can sort of see the muscle down there also, which is a V-shaped, which is V-shaped. So on Drosophila, we didn't use the adult animals, but the larvae, and we took third instar larvae, and we prepared them. We did a dorsal dissection. We sent them down first, which you see in the second picture. We did a dorsal dissection, and then you take out the intestines, and then you look at the body wall, which you can see in the fourth picture. And also, at the bottom of the picture, you can see the CNS of the fly. In the next picture, you can see where we inserted the electrodes. So on the left side, you have the sucking electrode to stimulate the nerves, and on the right side, you see the intracellular electrode coming into the picture and then going into the muscle to record potential. And also in the last picture, you can sort of see the outline of the intracellular electrode coming in from the right-hand side. So the question is, why is it so relevant when there is a change in pH? And it's actually a central topic, especially in human veterinary medicine, because there's this state called acidosis, when the body can't inhale or take up all the oxygen it needs, or not exhale properly and get rid of all the CO2 that it produced. So in the body, the content or the level of protons rises, causing an acidosis. So it can either be respiratory, like I just explained, or a metabolic acidosis. And in metabolic acidosis, there's just not enough oxygen for the organ to function, and this can happen during hypoxia or ischemia. So let me explain a quick background on the neurophysiology. What you can see here is a synapse, and there's a presynaptic terminal with the vesicles in there, and then there's the synaptic cleft and the postsynaptic membrane, which can be either a different nerve cell, just a neuron, or it can be a muscle cell. And so once an action potential, so stimulation, comes down the membrane, voltage-gated calcium channels open. And then this causes the vesicles of the membrane to fuse and let go of the transmitter, which diffuses into the synaptic cleft and then down to the receptor, where it can either open ion channels right away, or they can function via a second messenger system. And then, after they had their effect on the receptor, they're recycled and taken up into the presynaptic terminal again. So now we'll come to the quick basics of electrophysiology. So first I will explain the resting membrane potential, which is important because we looked at it to find out whether CO2 or pH had an effect on it. Then we will look at the spontaneous quantal events, also called minis, and then take a look at EPSPs, which are the excitatory postsynaptic potential. So the resting membrane potential is a potential every cell has, and it's due to the different concentrations in potassium and sodium inside the cell. So inside and outside the cell, actually. Because in the inside of the cell, the potassium level is very high, so it's up to 150 millimole per liter. And out in the outside, it's only 5 millimole per liter. So what the ions try to do is get out of the cell because of the chemical gradient. So once they diffuse outside, the charge inside the cell becomes negative. And so there's this chemical gradient leading them to the outside, but once those ions diffuse to the outside, there's this electrical gradient, which holds them back because the inside is charged negative, and those are actually cations and want to go back. So then there is the steady state when basically the potassium ions are in a state where they diffuse in and out at the same level because they have this chemical and electrical gradient. So what happens once an action potential comes? So the action potential is basically driven by the sodium current, because once the transmitter binds to the receptor, and if it's an ionotropic receptor, then it opens ion channels such as sodium channels. Then sodium is very high outside the cell but very low inside of the cell. So the sodium diffuses inside and depolarizes it, because it's usually charged negatively in the inside of the cell, like we just learned, but once the sodium goes inside, it's charged positively in the inside in comparison to the outside. So after that happens, then we can see the depolarization in the membrane potential. And then the the sodium channels are inactivated, and potassium diffuses out of the cell again, because now the inside is charged positive. So the positive potassium, so the cations, diffuses outside of the cell again. Usually there's something you call an undershoot, because a lot more potassium goes outside, so there's a slight hyperpolarization, like you call it, but then once all the currents are closed again, you have the resting state again, so the resting membrane potential again. So now let me come to the spontaneous quantum events. So spontaneous quantum events are just as a spontaneous release of neurotransmitter without a stimulation of the synaptic terminal. So there's no action potential actually triggering it, and so it's only a small number of vesicles fusing with a presynaptic membrane and then releasing transmitter, and then this binds to the receptor, and you can see a slight depolarization at the postsynaptic membrane. And this is also dependent on the intracellular calcium concentration of the presynaptic terminal. The last thing I want to talk about here is the EPSP, the excitatory postsynaptic potential. This is a potential that is usually a lot higher than the minis we just talked about, and so you can measure it after the stimulation of a nerve. And the depolarization is just a lot bigger than in the minis. So once the action potential comes to the presynaptic terminal, it triggers the vesicles to fuse with the membrane, and then you have a lot of transmitter diffusing over the synaptic cleft, and then you have a stronger depolarization at the postsynaptic membrane, which you can also see in the second picture. On the x-axis, you have the time in milliseconds, and then on the y-axis, you can see the current, so you can see an actual depolarization right there. So now I'll explain the way we did our experiments. It's basically really simple neurophysiology, because you have the motor axon, so the nerve, and you stimulate it, and the motor nerve ends at the neuromuscular junction to muscle fiber. And then we put an electrode inside the muscle fiber and measure the potential. So now the question is, what does pH do? And we wanted to test what happens when the pH is lowered. So there are different methods you can use to lower the pH in research conditions. The first one is the ammonium pre-pulse method. So you use ammonium chloride, and this dissolves in water to NH3 and H+. And NH3 can easily cross the membrane and diffuse inside the cell, and then it takes up a proton again. So it's basically alkalizing the inside of the cell for the moment. So we see it's taking up the proton, but now the cell starts to work against this because it wants to keep the pH stable inside. What happens is that it's taking up protons in exchange for sodium. But once we wash out the ammonium again, the ammonium chloride, and the NH3 diffuses out the cell and leaves the proton. So here we have an acidification of the cell on the inside. So this is one of the methods, which is pretty neat. And then we have easier ways to do it also. One is propionic acid. So propionic acid is just a simple acid that diffuses over the membrane, gives off a proton, and then acidifies the inside of the cell. And the last method you can use is the CO2 method. You just take CO2 and put it in the water, and it will diffuse over the membrane because it's quite lipophilic. And then it's broken down into bicarbonate and proton, and the proton by the carboanhydride reaction. And what we also found in our studies that the CO2 must have some effect on the postsynaptic cell, which I will explain to you later during the explanations of our experiments also. So let me come to our results now. The first thing we looked at was the resting membrane potential. And we used the ammonium pre-pulse method to do that. And so first, the crayfish was put, or the preparation of the crayfish, rather was put in the saline and the resting membrane potential was looked at. And you can see here that it's about minus 80 millivolts. And then we changed the saline for a saline that was low in sodium, because later on we were going to add the ammonium chloride and then increase the osmolarity again. So we did a low sodium solution, because when we added the ammonium chloride, it was then isoosmolaric. And then at the end, we exchanged it with the low sodium saline again. So on the y-axis, you can see the resting membrane potential, and on the x-axis, you can see the time and what we did at each step. So what you can see is then when you add the ammonium chloride, which is a basic alkalizing solution, you can see that the resting membrane potential depolarizes. But once it is washed out and the pH is lowered, you can see that it repolarizes again. And now we looked at the amplitude of the EPSPs. And the amplitude you can see on the y-axis, and on the x-axis you can see the time in seconds. And each dot here stands for preparation we did. So first, in saline, you can see that the amplitude of the EPSP is about 20 millivolts. When you add the low sodium, it doesn't change really, and when you add the ammonium chloride, it doesn't change either. Once you wash out the ammonium chloride though, and it's acidic inside the cell, you can see that the amplitude of the EPSP rises a lot. But it's only transient, so after a certain time it decreases again also. And so we did the same thing basically with CO2 in comparison to the ammonium washout method. So here again you can see the amplitude of the EPSPs on the y-axis, and you can see the time on the x-axis. And here again each dot stands for preparation. So first in saline, you can see that's basically at the same level, the amplitude. The scale is a little different here in comparison to the left graph, so don't get confused by that. It's only going up to 20 millivolts, and on the left side it's going up to 100 millivolts, so you can't compare it exactly. And so after a certain time we added the CO2, and what you can see is that the amplitude in EPSPs decreases a lot. So in comparison to the ammonium washout, where we have an increase, here we have a decrease. But what you can see is that this effect can be washed out. So after we wash it out, we have the same amplitude as in the beginning again. So here you can already see that maybe CO2 and pH have a different effect on the cell. So here's the same thing again, and shown a little differently. So we look again at the 10th EPSP, so at the excitatory postsynaptic potential. So with CO2 and A, we see that we see it in normal saline. So that's just as a reference. And once we add CO2, we don't have any response. And then we added CO2 and glutamate, and then we didn't have any response either. And actually in a physiological condition, once you add the glutamate, you should have a response, because that's the way it usually works. And in the last graph, in D, you can see that we can recover the potential, because once we wash it out, we have those EPS, we can see the EPSPs again. So if we look at ammonium chloride in comparison though, at 60 millimolar, we see that in saline it is quite normal, because that's our reference. And then when we lower the pH with ammonium chloride in B, we still have a response in comparison to CO2. And when we look at C, we have a depolarization at the very beginning, but then we have no response. And this is the graph for the ammonium chloride and glutamate. And usually you would think that in a physiological condition it should depolarize, but it did depolarize at the very beginning, but then there's no response because the channels for the receptors for the glutamate are desensitized and thus give no response anymore. And in the last picture in D, you can see that also here a recovery is possible, so we have the normal amplitude of the EPSP again. So once again here you can see that CO2 and maybe low pH by the ammonium washout show quite different responses. Now we'll look at the Drosophila as a second example, and also here we look at the resting membrane potential. So here on the x-axis we have the time again showing the different steps just like we did in the crayfish. And on the y-axis we show the resting membrane potential again. Each line here stands for a preparation we did. So what you can see here is also that the resting membrane potential differs a lot. In Drosophila, that's the problem we usually have, but it's around -50 millivolts. And once the cell alkalizes, the resting membrane potential depolarizes. And once it acidifies, it repolarizes again, but not to the level it initially did. So this is a little different from the crayfish here also. So what we looked at next was the resting membrane potential at different concentrations of pH and CO2. So I will talk about the first graph at the beginning. So we have the resting membrane potential on the y-axis and the time on the x-axis, as usual. So first we have a pH of 7.2, which is the normal pH we usually use. And we have a resting membrane potential of about -60 millivolts. And with the ammonium washout, we get a pH of 5.0 in the solution. And you can see a slight depolarization. And once you add the glutamate, you can see that it really depolarizes and the resting membrane potential goes up a lot, because that's a physiological condition, just like I explained it. Once we go back to the pH of 7.2 though, we can see that the resting membrane potential is going back. So it's going back and repolarizes again. So we can wash out this effect. When you look at the resting membrane potential concerning the CO2, you can see that at pH 7.2 we again have this resting membrane potential of about -60 millivolts. Once we add the CO2, we have a depolarization here. However, once we add the glutamate and the CO2, we do not have any response in comparison to the glutamate and the low pH up in the first graph. So here again, we have this difference in response with CO2 and glutamate and the low pH and glutamate. And at the end, you can also see that this effect can be washed out, and there's a recovery in a way possible, because at pH 7.2, we have this resting membrane potential of about -60 millivolts again. So if we look at the amplitude of the EPSPs like we did in crayfish, we can get the following results. On the x-axis again, the time, on the y-axis, the amplitude of the EPSP, and the ammonium washout method was used. So in the normal saline or the hyperosmolaric saline, you can see that the EPSP is about 20 millivolts. Once it's basic, it's going down, so that's a little different from the crayfish. And once it acidifies, it goes up first, the amplitude, and then drops again. So that's different also. So if you look, so we have this one, a different graph also compared to CO2. And so on the x-axis the time, on the y-axis the EPSP amplitude. And here in the first part of the graph, you can see the EPSP at a pH of 7.2. And the squares represent the press we did with CO2, with a change to CO2, and the dots represent the press we did with a pH of 7.2 to a pH of 5.2 with ammonium chloride. So once we add the ammonium chloride, you can see here the dots that the EPSP amplitude decreases. so the pH decreases the amplitude. And if you add CO2, though, it basically shuts down the amplitude there, so this effect is even more severe. But in both cases, you can see in the third part of the graph that there's a recovery possible. So what we also did is look at the minis with propionic acid, though. And this is a recording you usually get once you put the electrode inside of the cell. That was the moment when I had to leave the lab, unfortunately, so I don't have any specific data on that yet. So if you look at it, you can see that on the x-axis there's the time. Each number here stands for about a second. And here on the y-axis you have the amplitude of the EPSVs. And in saline you have of the EPSVs, of the minis. And here you can see that there are quite some minis, usually in saline, in the fly. If you look at, if you add propionic acid, though, you can see that the minis increase a lot. And now we wanted to know whether the calcium from extracellular had an effect on that, because for the minis to increase, there needs to be an increase in calcium inside the cell, because like I explained at the very beginning, it is basically there's basically, well, it's dependent on the calcium concentration on the inside of the cell, how many minis there are. So a potent calcium blocker for the voltage gate and calcium channels is cadmium. So if you add cadmium just to the normal saline, you get a strong decrease in minis, as you can see here. But if you add propionic acid and cadmium and put them together, you can see that they're still there. So you still get a response with minis. So somehow the propionic acid also has to facilitate the calcium release from inside of the cell. So what I just explained, you can also see in this graph. So on the y-axis, you can see the number of minis in 10 seconds. And then on the x-axis, you can see the time. And first we did it in saline, so that's a reference. And each point stands for preparation. Then we did it in cadmium, and you can see that the number of minis decreases. And then at the very end, we added cadmium propionic acid, and you can see that there are still some preparations where the number of minis is increased, and therefore you can see that the calcium has to come from intracellular to release those minis. So to sum it up in my presentation, first of all, is that CO2 does not only have an acidifying effect, but that there must be different action mechanisms of action for CO2 and pH, and that the neurotransmission is blocked by CO2, and so CO2 must have some effect on the postsynaptic cell. So if you acidify the cell, there's a depolarization of the resting membrane potential, and there is an increase in EPSPs by acidifying the cells in crayfish, not in fly cells. And there is a higher excitability of the cell if you add propionic acid, and propionic acid promotes the release of calcium from the inside of the cell and not from the extracellular room. At the very end, I would like to thank Dr. Robin Cooper, who was my mentor in the University of Kentucky and helped me to do all the experiments and taught me a lot. Also motivated me to attend the Neuroscientific Congress in Göttingen this year. Then Paul, who did all the graphs, Hannah, who worked in the lab also, then Professor Einspanier, who is my professor who wrote the letter, or was my reference for the scholarship program, and then Professor Geber, who is giving me this opportunity to give this presentation here today. those are my sources. And then I would like to thank you all, and if there are any questions, I'm open to discuss them. Thank you.